Ready when you are.
Okay, great. So welcome everybody to the Longevity Biotech Show. Here is where we interview scientists, entrepreneurs, and thought leaders advancing the state of the art in longevity biotechnology and how it's going to impact us and the world in the decades directly ahead. Today we have Jean M. Hébert, hopefully I'm pronouncing that correctly. Very interesting professor and researcher at the Albert Einstein College of Medicine in New York, if I'm not mistaken. We have, I guess, scheduled till 5, but Jean said that we might be able to go a little bit past that. So I guess what we'll do this time is we'll have a few minutes of intro, maybe 15 minutes of prepared questions from each of Nathan and myself, and then maybe 15 minutes of audience Q&A, and then if Jean has time, we can go past that, past our excuse me, scheduled cutoff time at 5. So with that, how should we do this? Nathan, you want to go first, or I'll just do a quick intro here?
Yeah, let's just do quick introductions. I'll introduce myself. So I'm Nathan. I'm the founder of longevitymarketcap.com and the writer of the longevitymarketcap.com newsletter. It's basically a once a week sort of roundup of all the developments in the longevity industry. And I'm also the founder of longevitylist.com, and that's a place where you can find jobs, companies, and investors in the longevity space.
Jean, you want to give a quick intro? I mean, we're going to be talking about you the whole hour, so whatever you usually do as an intro here.
Oh, sure. Yeah, so I mean, as you mentioned, I'm in New York at the Albert Einstein College of Medicine. And the research we do is cell replacement aimed at repairing and rejuvenating the brain. In fact, what we're really trying to do is reverse age-related damage in the brain, ultimately. And so I'll tell you more about that during the conversation, I guess.
Great, and I'm Robert. I'm an aging biology researcher currently with the Cohen Lab at the University of Sherbrooke, short distance outside of Quebec in Canada, outside of Montreal in Quebec, Canada. I have about 10 years of professional experience as a bioinformatics and research software engineer in academia and industry. So, Nathan, you want to start with the questions here, or because you actually invited Jean to the show here, so and you read his book first, so maybe it'd be better if you started.
Yeah, totally. I'll take it over from here. Yeah, so it's a great pleasure and honor to have Jean Hébert here. I read his book a couple months back, and it was just like a total eye-opener and very interesting. Some really big ideas that I'm really excited to dig deeper into. So maybe, Jean, you can just start off by telling us a bit about your backstory. So where does your longevity story begin?
It's a long longevity story, I guess. I've been interested in this since I was a little kid, and realized that we fall apart as we get old. And I thought, well, maybe there's something we can do about this, and that sort of guided my interest throughout my schooling and my career. So initially, I was interested in genetics because at the time, that's sort of the code of life. And I thought if there's anything we can do, it's from a genetic perspective. So I got my Ph.D. in genetics, and then realized, genetics is a real useful tool. But in terms of directly addressing aging, regenerative medicine showed a lot more potential. And so I've sort of been focused more on that. And regenerative medicine is a huge field where you have to specialize for different parts of the body. So I thought, well, what's the most important part of the body? and that's the part of the brain that we use for our conscious thinking and everything. I thought, and I guess some people would agree. And so I focused on that, and how can we reverse the damage that occurs with time in this part of the brain. And so that's what we're actively working on now.
Yeah, that's super interesting about the brain, and I think that's really the main focus about replacement strategies. Because when I first heard about your book, I saw the title, Replacing Aging, and I was like, oh, okay, I know what this is going to be about, organ transplants, xenotransplantation, lab-grown organs, and that kind of stuff. And I was thinking to myself, oh, but, this will never, solve the problem of the brain. You can't just replace the brain, right? So, but then when I actually opened up your book and started reading it, I was like, oh, this guy's actually trying to replace the brain too. That's crazy. But maybe you can just tell us a little bit about that strategy, right? Like there's replacement of the body, right? And there's various approaches to going about that. But the brain is just like one of those organs that most people think can't be replaced. But you have an interesting approach to doing this. Maybe you can explain.
Yeah, and thanks, for your questions and having me here too as well to have the opportunity to tell you a little bit more about this. Um, so yeah, the book is sort of divided into three parts. One of them is, like you say, how we can actually um, progressively replace the brain, which is somewhat counterintuitive, but I'll say a little bit more about that to explain why it's possible in theory and in practice. The other two parts of the book, I don't know, maybe we'll touch upon them a little later, but is sort of um, some of the limitations of the other approaches, and why I think they're more focused on the effects of aging rather than the core, the essence of what aging really is. Um, and then, I talk about other parts of the body as well. But coming back to the brain, uh, the reason why it makes sense to think about replacing the brain progressively over time is that's sort of the nature of the brain to start with. It's very plastic, and we've known this for a long time in the neurosciences, where uh, experiments have been conducted with model animal systems showing that uh, parts of, in particular, the neocortex, which is what we use for our most advanced cognitive functions, can be repurposed over time for different uses, depending on whether we no longer use needed the previous function it was used for, or whether we need it more for a new function. So the neocortex is very plastic, and the substrate for who we are in terms of our memories, our thought patterns, is continuously changing over time, and can actually change a lot faster than it normally does in our daily lives. And one of my favorite examples, there are multiple, but is the example in humans of slow growing benign gliomas that occur in the language center. And these gliomas, over the course of 5 or 8 years, can completely eat away at the language center so that the original language center is gone. And eventually, the person with this tumor gets a bad enough headache or, a seizure that they go to the hospital, and they're told, oh, bad news, you have a brain tumor. Good news, it's benign and we can take it out. So they take it out with a little surrounding tissue for good measure, which is what they always do to try and avoid recurrence. And the patients can still speak normally, even though you can clearly see on an MRI that they have a big hole where the language center should be. And so when they look at where the language center has moved to, it's moved to a new part of the neocortex over time. Um, and this is very different, for example, than what happens in a stroke. So they actually, in their study, do a side-by-side comparison with same aged individuals with the same amount of damage to the language center, but due to a stroke, which is a catastrophic event where individuals don't recover any language, maybe a few words, but it's very minimal. So these are very different outcomes to essentially the same amount of damage. And what's different is the time that it took for the damage to occur. In one case, stroke, catastrophic event, no time for plasticity, the brain to move the function to a new substrate. Whereas with a slow growing benign glioma, there was 5 to 8 years for language to move to a new part of the neocortex. And this level of plasticity, where a function as complicated and as dear to us as language can move to a new substrate, tells us in principle that the brain can be replaced progressively over time, right? And so since the book, actually, we've elaborated strategies that can be implemented, that we can test at least, to show that this can actually be done. Um, but the other reason why, uh, in practice, not just in principle, but in practice, we think that the brain can be replaced is somewhat more recent evidence, putting in where lots of labs are doing transplants of immature neocortical precursor cells and other areas of the brain as well, and showing that these immature precursors behave like they do in a developing individual, as opposed to an old adult individual, meaning that they project, they differentiate normally, they project to their normal targets, and they become functionally integrated in existing circuits. Um, so those two things together tell us that it should be possible to, using replacements, reverse brain aging by progressively replacing the different parts. There's a lot of things that need to be worked out to make that happen. This is a very big project. I don't mean to make it sound like it's simple. It's very complicated. There are a lot of cell types to consider. There are a lot of things that need to empirically be tested uh, to make sure that they work. But there's no biological reason why we can't reverse brain aging. The reasons right now are just technical. So I think we'll be able to overcome them.
Yeah, I really like that point that really this approach is fundamentally sound. It's really, as you said, a technical problem. It's like an engineering problem.
Yeah.
Whereas if you try to reverse, aging in the brain with other means, maybe like with a drug or something like that, maybe you would have to understand the entire system before you could even begin to think about, okay, we'll try, this drug or this combination to try and reverse aging. And yeah, so I just really love your approach because of its fundamental soundness. But moving on to just your idea, so we have uh, this hypothesis that we can replace aging, or sorry, reverse aging in the brain with replacement. What does the roadmap look like? Like, what sort of technical details are you?
Well, so again, if we just focus on the neocortex to start with, because it's sort of the biggest part of the brain, it's the outer part of the brain that's all folded. That when you think of a human brain, that's the image you have in your head, right? So it's really the biggest and most important part of the brain. So if we focus on that to start with, but eventually not exclusively, because the other parts are also important and will need to be replaced. The neocortex is composed of multiple cell types. For the most part, they're very well defined. The cytoarchitecture and the arrangement of those cell types is also well known. So one of the first big steps is to be able to reassemble those cells in sort of this young, immature tissue where the ratio of cell types is what it's supposed to be. The relative maturity state of the different cell types mimics what they are during normal development, because we want this tissue to mature as normally as possible. And the sort of topographical arrangement of these precursors also matches what it should be during development, again, just to foster normal development. So, that's one of the big technical questions that we're starting to tackle. But again, there's many moving parts that have to come together to make this work. Um, Another one I think is really important is just to show proof of concept that this approach works. And that's something we're also developing an assay for. Um, so basically recovering a lost function with a grafted tissue. And the tools are available now where we can specifically silence transiently the neurons in our graft without affecting anything else. And if we can do that, then we can show that the behavior that we think this graft is now um, allow underlying, we can transiently silence the neurons and show that that behavior can no longer occur, and then reactivate that graft and show that the behavior comes back. Then we've pretty much proven the concept that we can do it. So I think that's a very important experiment. So we're working really hard on that as well to get that to work.
Right. So these experiments will be done in mice first, I'm guessing, right?
Yeah, we start with mice and then move from there. I think
Okay,
we can
great.
go directly from mice to humans. We might have to do a larger mammal, but yeah, that's the idea.
Okay, cool. And are you looking for collaborators? Are you hiring people to help you on this project or looking for funding? Maybe if you want to speak to that.
Yeah, absolutely. It's a little bit. I mean, we can hire a couple of people now, but it's really funding first, then people. So yeah, I've been actively seeking funding more and more so from the private sector. Uh, this is a little too risky for NIH funding, for example. They tend to be very conservative in the projects that they like to fund. It's also a very big project, like I said, and so it does require more funding than most of the NIH funding mechanisms would allow. So that's why I've been particularly looking at alternative funding mechanisms through the private industry. And once that comes in, then yeah, we already have a team of scientists. These are heads of labs who have agreed to participate in this project um, by participating in the hiring of the specialists that we need, and if necessary, their training and overseeing the different aspects of this project to make it work. And there's a team of 15 of us. So really, first step is funding. Second step is then expanding these teams to be more functional.
Great. Yeah. So if there's anybody in the audience or any listeners out there um, who have access to funding and are very interested in Jean Hébert's program to reverse brain aging, yeah, you can go contact him. I think just by email or his website at Albert Einstein College of Medicine,
Yep.
unless you have another. Yeah, okay, cool. So let's turn it over to Robert now. I know he has a bunch of questions. So Robert, do you want to take over?
Yeah, thanks, Nathan. So, Jean, very, very interesting. So I confess that first of all, thank you, Nathan, for inviting Jean and discovering his book and work and all that kind of stuff. I wasn't aware of it really until you brought him up. So thanks for that. So, Jean, I confess that I know we've been emailing a little bit before the call here, but I actually just finished reading your book a little over an hour ago. I caught up very early today, and I went through the whole thing. And it's, if I may say, it is one of the most thought-provoking and, in some ways, imaginative books that I've read since Eric Drexler's Engine of Creation, which, in my view, that book is better than science fiction, or better than a lot of science fiction. So kudos to having the courage to write such a book and discuss your ideas and all that. I have way too many questions for the few minutes that we have in the call here, and we do want to try to open this up to the audience. We have a number of people here that might be very interesting to add to the conversation. Like I said, we have Colin Ewald from the ETH, who we had on, was it like a week or two ago? I'm losing track of time here. I did want to ask. I'm looking through my notes here. I took extensive notes on your book. So, by the way, I'm just going to say again to everyone listening, this is an exceptionally good book. It's probably one of the best books discussing the prospects of reversing or replacing aging. In some ways, it's even better than the book that Aubrey wrote, because it doesn't get quite as technical for lay audiences, and it really just focuses on the main points. So I got to limit my questions, and I didn't have too much time to sort through everything. But all right, let me start with one major point that I think may be worth expanding the conversation to even perhaps another call, which is, like as you said earlier, you spent the early parts of the book, one or two sections, discussing how approaches to dealing with age-related damage that are not involving replacement of the components of the whole organs are not likely to work. And you mention in particular at one point how there are people in the field of aging biology and longevity therapeutics that are working on, for example, telomeres and lengthening telomeres, or mitochondria and reactivating or restoring their function, amongst other things that you mentioned in the book. Can you elaborate a little bit on, just for the people here that aren't so familiar with what you've said in the book, why you think that those approaches are not likely to be as effective as what you're suggesting?
Yeah, no, thanks. Yeah, I'm glad you asked that question. So I think there's a very big blind spot in the field of longevity research now. And it relates to the fact that I think people are ignoring what aging is at its core, and instead are focusing on, one or many of the effects of aging. I think the most useful definition and the most accurate definition of aging is the accumulation of macromolecular damage that occurs over time. In all animals, not all, there's a few exceptions, but most animals, and in all our tissues. And this is important, and we've known about this damage accumulation for decades from biochemists, but somehow it's really largely ignored by the field. If, for example, there lots of proteins have been shown to accumulate damage over time and have really bad effects on the function of cells. But let me give you a couple of examples to try to make it more concrete. One is elastin. It's a protein that's found in the walls of blood vessels. It's made when we're developing, as embryos, fetuses, and maybe still growing as children. After that, it's not made anymore throughout life, and it accumulates damage. So it's well documented. It's actually a wonderful biomarker that is also completely ignored in this chase for biomarkers. But it accumulates damage over our lifetime to the point where when we hit 90 or 100, this elastin protein is really not functioning well at all. It's not doing what it should. It does not allow blood vessels to contract or expand at all. And so and blood vessels are throughout our body, right? So any approach that doesn't take this kind of damage into consideration in trying to reverse it will likely meet a wall of for longevity, right? And this was actually proposed by a couple of groups again decades ago that this accumulation of damage, if not addressed, sets a limit to longevity. right? Because these proteins are not turned over. You can change the epigenetics of cells. It won't matter. They're not going to replace the elastin that's non-functional that's there. You can change the metabolism of cells. You're not going to remove the damage to the elastin that's there. And I'm focusing on elastin, but it's true for a lot of collagens. It's true for even some intracellular proteins that appear to be exceedingly stable. There's components of the nuclear core complex that don't seem to turn over. Some components of the nuclear core do, but the scaffold proteins in the nuclear core complex don't seem to turn over, and they're known to accumulate damage over time. Um, so one might think, okay, well, so a lot of these approaches that are very popular now don't address this damage. But maybe there are other approaches other than replacement that might work, like reversing this damage or undoing at a molecular level this damage. But elastin and these nuclear pore complex scaffolding proteins and long-lived collagens, are just some examples. But there are so many. I mean, pretty much all proteins accumulate damage with age, and one of the reasons is that some of these forms of damage are not easily dealt with. So we have all sorts of proteases that normally turn over proteins. So you would think, oh, that's good. You can just get rid of damaged proteins and they turn over. Well, one, as I mentioned, some proteins don't turn over. Another thing is that um, uh, that the damage that these proteins incur inhibit the degradation by these proteases, so that they just accumulate. So you get accumulation of damaged proteins, even ones that normally turn over more quickly. And then the forms of damage. I mean, we hear about glycation, oxidation, so different forms of cross-linking. There's also racemization, where amino acids go from the L conformation to the D conformation. Also a very good biomarker over time. It's very linear. But there's many others. Depurination, depyrimidination, forms of aggregation that we hear about, deamidation. I mean, the list goes on. And these forms of damage occur differently in different species of proteins over time, right? So trying to imagine how you're going to undo this, and this is only protein. So this type of damage also occurs to lipids, our fats, to carbohydrates, that are ubiquitous in the body. Um, and of course, also we have different forms of damage to the DNA, which in itself, is information content and irreversible in that sense, because it's also it's sort of this stochastic damage that requires information content to be repaired. So when you start putting all this knowledge that we have about damage accumulation, and you leave that out of the equation when you're trying to address how you're going to beat aging, I think we have we have to consider using replacements as a means of addressing aging. So if you replace a tissue, you replace all forms of damage in one shot, right? You basically set the clock back to zero. And if you replace enough of the body in a short enough period of time, you will become young again, right? I don't think anybody argues against that. What people are not necessarily aware of is that uh, regenerative medicine has made tremendous strides over the last few decades in replacing organs, in some cases in patients with lab-grown organs, and but for all organs, they're all in preclinical trials, and progress is constantly being made. Not fast enough, in my opinion. More funding needed for that as well. But that's certainly coming along. And as Nathan pointed out, it's when you do think about, oh, replacement would be great, but if you can't do it for the brain, what's the point? Uh, so the point I make in the book as well is that you can do it for the brain. And so I think I think it's a matter of time before we actually beat aging. But I think for many of the approaches that are favored now, they'll be met with disappointment or at least limitations into the benefits that they have. I think a lot of them acknowledge, a lot of the researchers working on these points acknowledge the limitations and are now talking about healthy aging, or uh, not getting the diseases of old age and yet still aging, right? Um, so I think that's a great thing as well. If you limit the amount of disease you get when you grow old, that's great. So that all that research might have some benefits, but in terms of actually impacting the aging process itself, or beating the aging process, I think we have to acknowledge that they will meet with limitations and maybe disappointments for those who think that these approaches might extend human lifespan well beyond 100 years. Um, so thanks for the question.
Well, thanks for the detailed answer there. There's, if I can just ask you just give a very short answer to this, to what you just said there. Other people in the field, do they call you a pessimist for saying that they might that these more conventional approaches are going to lead to disappointments or something?
um, I've gotten much more positive response than negative in that sense. So, because really what I'm saying is that we can do this. We can beat aging. I think it's inevitable. So I think, and I don't know if that's optimism, but um, it's a positive outlook, I think. Um, I just want to mention, though, for anybody who's interested and is not convinced by what I said about damage, a really good starting place is a review written by Alexander Fedintsev and Alexei Moskalev last year in June. That was published in Aging Research Reviews. It's one of those articles that anybody interested in longevity should take a look at, because they do a good job. They're probably more diplomatic than I just was in explaining why ignoring accumulation of damage is going to lead to disappointment. But essentially, the message is there that damage actually causes a lot of what are now considered the hallmarks of aging, are actually consequences of accumulation of damage, and they make a really good case for that.
Okay, thanks for that, and thanks for that recommendation. I'm going to definitely look that up. I've heard those names before for sure. Since we're short on time, and it'd be good to get some people in the audience here. My next question, to sum up your point, if I can, on page 186 of the book, you say quite succinctly and clearly that, in your view, the molecular and cellular damage that accumulates with age as a normal byproduct of life-sustaining metabolism is simply too integrated and complex for drug-based strategies. Probably that would be the single summary message that sort of captures everything that you say in the chapters that are dealing with why the non-replacement based approaches aren't really going to work as effectively.
Right.
My next question was going to be about something you said again, I'm checking my notes here. There's so much stuff. On page 160 of your book, this is the physical copy of the book, you say that for the brain, which cannot be replaced as a whole, special consideration needs to be given to replacing the extracellular matrix. To some extent, this problem is mitigated by the arrival of new replacement cells, since the young cells will create their own new and undamaged ECM. However, residual old ECM would have to be completely eliminated at some point during the rejuvenation process to truly cleanse brain tissue of age-related damage. At this time, the best way this can be accomplished has not been determined. So at some point in the Q&A, I'd like to invite Colin Ewald to follow up on that with you. But I do want to ask a very important question on my part. One thing that you touch on in the book, if I could just say to anyone that is going to read this book, especially people that are not as familiar with the truly far-reaching imagination of what is possible with technology, it may shock you. Some of the passages and descriptions of the book may shock you. There are descriptions which some people would describe as you know you use the Frankenstein example for one. At one point, you even have a chapter of that title, right? So, and you do say at one point in the book that you're not. How did you put it? You said you are not an expert by no means. Am I an expert in the social sciences? You said. And yet, you say that human beings and society are relentlessly adaptable, and so there's no inherent reason to imagine a population that lives longer cannot form a prosperous society. The question I have to you is, how do you address the folks out there, and there's going to be a lot of people out there who are going to be uncomfortable with, possibly scared by, and outright antagonistic to the kinds of research and proposals that you make in the book, like have, because you do touch on this in the book, but don't really address it very much, aside from mentioning that it's gonna, it's a concern. So how would you elaborate a little bit, let's say in a minute, so we can bring some people up on stage from the audience, as to how you intend to, or how, in your opinion, the industry as a whole should address those people, because there are going to be people like this out there who will say these things.
Yeah, I mean, again, there are other people who have, like Aubrey, for example, and others who have spoken very eloquently about um, how you address some of the concerns that people bring up in general about extending the human lifespan. Um, so I don't know if you're asking more specifically about the approaches that I'm suggesting, about that involve replacements. But I would say this is already happening. Um,
Perhaps I can be a little bit more clear. Also,
yeah,
not be as clear as you. I'm just gonna say it here. Some of the stuff that you write about in the book will be described as transhumanism, and there are people out there who are strongly against any kind of anything even approaching transhumanism. And like, you go right to the limit of what, at least in some of the chapters of the book, of what is possible. That's why I said it's as thought-provoking, as imaginative as Eric Drexler's book, as he talks about that kind of stuff as well. Now, the thing here is, not everybody agrees that the body and mind and brain are purely mechanistic, let's say, or largely mechanistic in the way that you describe, so that they can be replaced, piecewise, like you describe in the book. There are spiritual people out there. There are people who are not as much in favor of advanced technology as many of the folks who are advocating, anti-aging strategies. How do you address those people? Because what if there are people out there, and there are people out there, who just don't want this kind of stuff, and they're happy with the way things are?
yeah. There's a lot actually in that question. The first point is transhumanism. I actually don't see myself as a transhumanist. I mean, I don't have any issues with the transhumanist movement, but the transhumanists are looking to fundamentally change the human nature to something better. Whereas, my whole focus is to maintain who we are, instead of degrading and aging and eventually meeting an inevitable demise. So, what I'm really trying to do is not transhumanism so much as preservation of humans as much as possible as they are. So that might be a little different. I don't know if that's necessarily more comforting to some people. As you say, some will be fundamentally against this, um, no matter what. But this has been the nature of science and technology and the development throughout humanity, right? I mean, people were against the bicycles because it was unnatural until, of course, automobiles came around, and then they forgot all about bikes. There's an adaptation period for all these things. Organ transplants, that was problematic at first, but they're pretty routine now from donors, and they're becoming more routine for certain lab grown organs. And, it's just part, it's all part, for me, it's part of the regenerative medicine aspect. All these are just doing good in terms of health and longevity. Um, so I, I think, yes, some of the approaches I touch upon in the book are definitely going to be viewed as a little more controversial. But, those are all possibilities. They're not the only approaches. But certainly in terms of some of these body part replacements, even with prosthetics, right? I mean, I just saw, last summer, somebody in Central Park here in New York running upstairs with a prosthetic leg, and movement looked totally natural. I mean, these things are getting really good, and so, but they're they're mimicking a normal leg, and so right now these things are all being used to treat injury and disease. But when they become more commonplace, which is happening every day, I think people are more exposed to them, become more accepting of them, and see that this is good. It makes us allows us to live our lives more fully for longer. So I agree. I know what you're saying, though. I mean, there's some people there's going to be pushback, and there always will. But there that's a big endeavor, something that we all need to be aware of. And if we're interested in longevity, we all need to be outspoken about.
All right, thank you so much for that answer. Maybe it's something to come back to in another call or
Sure.
an edition of your book or something. So since we're approaching the at least top of the hour here, let's open this up, invite some people from the audience. I forgot to mention earlier, we are recording this, so if you come up on stage, you're consenting that your image and voice is going to be part of the recording. So I invite, okay, here Colin's coming up to ask questions to Jean, and we'll try to get everyone in order. And John, if you can stay a little bit past 5, just let us know how much time you have, and we'll try to accommodate as many people as we can. So, Colin, thank you so much. What a surprise to have you here. Please, the floor is yours. Colin, you're.
Hi, Colin.
Are you there?
Yeah, can you hear me?
Yes, you were muted for a second.
Perfect. So, first of all, fantastic show. I always love come back to hear more, and I was really looking forward to the show with Sean today because I must say I'm a big fan, and Sean was kind enough to send me his book, so I'm super happy about this. And I can just reiterate, the book is fantastic. It's a very good read. It's thought provocative, and it's just excellent. So, Sean, I just want to ask you, of course, some question about the extra matrix. I mean, in the book, you already mentioned it, it's like a big part of the body. How you envision to replace, like, big extra matrix parts, connective tissue? That would be my first question.
Yeah. So
Just before, can I just say to everyone listening, Colin is a world expert in ECM research at the ETH. That's another thing. Maybe when you come up on stage, just say your name and where you're from. Might give some background to everyone else listening. Sorry, with that, go ahead, Jean.
Yeah, sure. No, I mean, Colin, yeah, you're absolutely right. The ECM is a major part of the body, right? Collagen, extracellular protein. I mean, it's a family of proteins, but it comprises 30% of the protein in the body. So yeah, it can't be ignored. If you're thinking of aging, you have to think of the extracellular matrix. In fact, the aging extracellular matrix uh, has been shown to be the causative reason, or at least one of the causative reasons for stem cell exhaustion, for inflammation, for cell senescence. So yeah, I mean, it can't be ignored. So the way that we propose replacements for most of the body is not so much an issue because you replace the cells and the extracellular matrix at the same time. For the brain, in the book, I was more open to approaches, but over the last, I probably wrote that 2 years ago now. So over the last, and it was published last November. But I think really there's a much clearer path to success in replacing brain tissue, and it also involves similar to what happens with a slow-growing benign glioma, where you have obliteration or silencing over time of one area. I think if we do that, as well as provide the new substrate, the transfer of function will occur seamlessly, as we know happens already. And then once the tissue is dead, that has been silenced, I mean, not biologically dead, it's still metabolically active, but neuronally, electrophysiologically dead, then it can be removed. And with that, you remove the extracellular matrix. And with the new tissue, you provide a whole new extracellular matrix along with new cells, pristine new tissue. So I hope that answers the question. That's sort of what we're thinking.
Yes, thank you. That really answered the question. Can I ask a second question? Just because to stay in this route. So there are experiments done taking in rats leg muscles from a young rat and transplant the muscle into an old rat, and vice versa. You can take an old rat muscle and transplant that in a young rat, and the old muscle functions like a young muscle. But vice versa, if you take a young muscle, place it in an old rat, the muscle functions like an old muscle. So aren't you afraid that replacing some organs and some tissue is just going to be basically old, even though they don't have the accumulation of damage?
Yeah, I mean, that's a very important question, right? And one that comes up. And so I think the muscle is one example, but there are a lot of examples like this where, you can take old cells, put them in a young extracellular matrix, and they behave like they're young again, right? So again, why what you're working on, the extracellular matrix, is really important and shouldn't be ignored, and vice versa, taking young cells and putting them in an old environment and they don't behave well. They behave like the old cells in the old environment. So there's examples of that actually for multiple tissues. And so the approach we're taking, though, is that we're replacing enough of a tissue, piece of tissue at once, and all the cell types in that tissue. So all the support cells, the vasculature, everything is young, surrounding our neurons, for example, for the brain. So it's still something that we would have to keep an eye on, the performance of the grafts. But so far, what we've seen in tests in animal models is that these um, these new cells behave like young cells. Um, and there's very limited evidence of this in humans because it hasn't been looked at, except one case in Parkinson's disease, where they looked at postmortem tissue and showed that the cells that they transplanted were, for the first, at least decade, devoid of the pathology that was seen that the neighboring, recipient cells were exhibiting. Eventually, they do succumb, but those are purified neurons as well, neural precursors that were put in. So the way we're doing it is going to be even better than that. So we would hope that there's, at least more than a 10-year, uh, um, you know, um, time when the tissue is not exhibiting any of the influences of surrounding aging tissue. But at the same time, we're doing progressive replacements to eventually get rid of all the old tissue. So, there might be a timing, a rate of replacement that will need to be determined. But I think it's totally overcomable. This problem of the old environment will, yes, eventually, win out. But if you're outpacing it, I think you'll be okay.
Makes really sense to me. So thank you very much for your explanation, and
Yeah,
I
thank.
really look forward to your fascinating research coming out of
Yeah,
your
I
lab.
hope we get to talk some more soon.
Thanks so much for that, Colin. John, welcome. Maybe a quick where you're from and your question.
Hi, yeah, sure. My name is John Young. I'm an MD, PhD with a PhD in artificial intelligence, and I'm looking to transition into the longevity space. And I also really enjoyed your book, Sean. And I actually had a question about an article you wrote in Trends in Neurosciences. I thought this might be helpful, but at the very end of the article, you say, at this time, there's no evidence that aging can in fact be reversed through cell transplantation. And I was wondering, you kind of touched on this throughout this clubhouse, but I was wondering if this still stands or if you have any updates.
Yeah, I mean, since again, that came out sometime in 2018. So his article was based probably on work done before that, up to maybe 2017. So I mean, the progress has been made in the last 4 years, and I've become a lot more confident that these replacements will work. I mean, as I was telling Colin and everyone else, in the book, I was uh, more open to different approaches, not knowing what might work best. But I think as we've examined the potential caveats and benefits and looked over the literature about what's known about um, the certain aspects of brain aging and and what's possible in terms of transplants, I think it's much clearer to me that now um, that we will be able to get this to work. I think I was being cautious in that review paper as well, but yeah.
So some of your more recent papers, or would there be, is there evidence or is there still no solid or I don't know, convincing evidence, I guess?
Well, so there's a lot of groups that have been transplanting precursor cells in the adult brain under different pathological conditions, and over the last 4 or 5 years, and it's reason for optimism. I mean, these cells do really well. They connect, they differentiate, they connect, they become electrophysiologically integrated into networks. If you put precursors in the visual part of the neocortex, for example, over the course of a month or a couple of months, they will behave like normal um, visual cortex neurons in that they respond, primary visual cortex, in that they respond the same way to um, um, the orientation of a visual presentation, as well as if it's a moving um, visual cue, respond the same way to a moving visual cue. Transplants to the motor part of the neocortex, if you excite those neurons, they will elicit muscle contractions. So, the evidence is just building that this is definitely, if not, I think it's more than worth pursuing. I'm confident that it will work.
Great, thanks.
Yep.
So
And
thanks for your question. So, Jean, how much more time do we have? It's a little bit past 5 here.
I can go on for longer. I don't actually have anything scheduled for the rest of the day. So.
So, Nathan, do you have anything now to add? I have plenty of questions, so I can continue. And again, anyone else, if you want to come up, just raise your hand.
Yeah, maybe I could ask a question. So, Jean, why hasn't Bill Gates reached out to you? Because, Bill Gates is a champion of Alzheimer's research, right? So I would think that the Alzheimer's community, or maybe the Parkinson's disease community would be really interested in this kind of research and this approach to potentially reversing these neurodegenerative diseases.
Yeah, I mean, that's a good question, Nathan. I don't know that Bill Gates knows that I exist, for one. But beyond that, no, there are a lot of people that could make a huge difference in accelerating progress in this area of regenerative medicine. Um, at least a dozen of them openly, pursuing the idea of longevity, and then, dozens more that are funding regenerative medicine in big ways. So, I mean, that's it. That's one avenue of funding that I'm trying to pursue, as well as these major philanthropists that can really make a difference. Again, because this is not a project that can be done. We really need teams of scientists for the different cell types. We're using, we need to optimize our methods for layering and topographically arranging those cell types. So we need very good tissue engineers. Um, we need electrophysiologists. We need systems neuroscientists. We need computational biologists. Um, we need all these people to come together, and that will only happen with some major backing. And so, yeah, I mean, if you have contact information for any of these people on your Rolodex, I'd appreciate it. I am sort of working towards that through other people who do have contact with these potential major donors. So it's a work in progress. Hopefully, we'll get there. Yeah.
Does that answer your question, Nathan?
Oh yeah, of course. Yeah.
Okay.
Thanks for the.
I guess Mihai, welcome, and then I'm gonna put a follow-up question to what Nathan said. Mihai, maybe a quick word about what your background is and what's your question?
Hey Jan, thank you so much for doing this session. It was quite interesting, and I must say I haven't read your book. I'm a software engineer. I don't really know that much about biology and such, but I'm very curious about the field. And listening to this, it just popped through my head that maybe some people would be open to having their brain removed and placing that into an artificial environment where it could continue to survive somehow. And I'm curious what you think about that. Like, is that something that would make the problem more tractable, or do you think that would be, to science fiction?
Yeah, no, I think that's a viable approach. I mean, I think that might be what Rob touched upon as as something that would be rather controversial, and I get a lot of pushback on. But I do mention that in the book, and there has been advances made in that respect as well. Right? We are our brains. Pretty much. I mean, again, not everyone would necessarily agree, but if I had one thing to preserve, it would be my brain. And there are systems that have been developed that can keep biological tissue alive for long periods of time, that could be optimized as replacements. So they perform the function of blood filtration, like a kidney would, of oxygenation, like the lungs do, as well as pulsatile delivery, like the heart does. Um, that could be used, right? So these systems, there's at least one example from a group in Yale where they've developed. I think, to my knowledge, that's the most advanced system. Um, it's not good enough to keep a brain alive indefinitely, I don't think, yet, but it's certainly possible. And then combine that with the ever improving brain machine interfaces, right? Where with artificial intelligence and these newer, very fine, minimally uh, invasive, minimally reactive, like carbon-based electrodes, like Neuralink is working on, for example. These interfaces can allow you to move, artificial limbs and everything. So, in a relatively short amount of time, these interfaces allow you to do that, and that's been shown in, quadriplegics already, that this works. So, yeah, you're absolutely right that the replacement of most of the body can be done with non-biological parts, and still allow us to be relatively functional, or perhaps even more functional. But I guess that's a little more, transhumanist. Um, so, Rob, maybe you're right on that point. I forgot I did discuss that in the book. But yeah, no, that's another possibility. Yeah,
Yeah,
thanks.
thanks for that question, Mihai. That's one of several things of that general nature that you discuss in the book, and that's what I'm saying. Like it's so that definitely crosses over into the realm of transhumanism, and um, it's something which you do mention as being of concern to some people, but like it would be perhaps a good idea to elaborate further that concern, and how to respect the wishes of people that are not interested in this kind of stuff, because there are plenty of people out there that are not interested. And also the interesting examples, like you gave an example of a religious doctor who nonetheless was pursuing some pretty out there experiments, in pursuit of this general direction of research back in the 70s or so. I got to look up my notes on your book here.
Yeah,
Um,
Robert White. Yeah, he's
Robert
the one
White, yeah.
who did the monkey head transplants, right? And he was developing this for humans, because he thought it was immoral, if we could save a human life with by giving basically a person or that would die from whatever degenerative disease a body that would allow them to continue living. That would be immoral not to do that. And so he was developing this, and yeah, he was very religious person. He was an advisor to the Pope at one time, um, and so these approaches are not incompatible with religious beliefs, certainly. So yeah, I mean that's another avenue that gets a lot of press in terms of being controversial. But again, it's to save someone's life. I think we can argue that it might be worthwhile.
Definitely something very, very interesting to come back to. So, if I can elaborate on maybe one of the questions I wanted to ask that sort of follows on to, Nathan was asking like why aren't people like Bill Gates looking at this kind of stuff, and also you mentioned earlier that a lot of people may not be aware of the progress in regenerative medicine and how much has been done in recent decades. On page 173 of the physical book, or the printed copy, you say, in general, predictions of success in research have been notoriously inaccurate, with the achievement of stated goals in some cases taking decades longer than expected due to unforeseen roadblocks, and in other cases being realized much sooner than predicted due to unanticipated technological breakthroughs. I put down this quote as, at least in my book, the most significant comment in the whole book, right? As relates to sort of
Thanks.
the trends that we are part of and witnessing, right, in our rapidly changing society. So I guess my question here would be, because you also say at one point, where was this? In a different page you wrote, when will we reach a stage when organs and tissues can be routinely replaced to cure aging? In 10, 20, or 30 years? It's hard to predict, right? So that's a very candid statement. I think that's something that needs to be said more often. It's hard to predict these things, and that's why I'm saying that the first quote on how some things happen much later than hoped for, and some things happen much sooner. So related to this question, I guess would be what are your thoughts on the technological and research trends in that sense, like of the speed of these things happening? And this, I guess, follows on to John's question, but also like what are some other developments of the kind of research that you're working on that may influence potential major donors if they say, well, this is amazing, but it could be used for this, and we don't want that, right? Or this is amazing. Have you thought about it being used for this? And we do want that, and then that might be easier to make the case if you say, like, look, we can actually use this same technology for this other area that you're interested in.
Yeah, I think that argument can actually be made pretty readily, and is made um, all the time in regenerative medicine, right? I mean, the progress that's being made, even though investigators are very well aware of implications in terms of rejuvenating or reversing aging, um, they're stated openly. Their goals that they state openly is to basically cure a disease or repair a traumatic injury, and so I think those arguments are made and they're readily accepted. Um, I think what will make a big difference in terms of the time it takes to get there, again, which is hard to judge, but what's clear is that there are a lot of things that need to be worked out technically, and if we need to work them out sequentially, it'll take a lot longer than if we can work them out in parallel. And what would allow us to work them out in parallel as opposed to sequentially is increased funding. Get more people working on that. So that's yeah. But I think in terms of getting the funding, I think it'll grow. I mean, I think this approach will catch on more generally. Regenerative medicine has always been a major interest um, to those interested in health in general, um, and as examples of lab-grown organs continue, then I think the interest will just grow, um, and if we can accelerate that, that'd be great.
All right, so many things to follow up on here, but we have Michael Weiss on stage. Maybe quick background on where you're from and your question.
Thanks, Robert. Hey, Jean. Super
Hi.
interesting. Yeah, I'm Michael. I run a large AI industry conference in the U.S., and I would say I'm an avid supporter of longevity. My question is, what is your kind of philosophical motivation behind your work? Is it, I'm Jean, I'm aging, I want to replace my whole body, or is it, this is just my greatest contribution? What's kind of your personal mission statement?
Mission statement. Well, my mission statement is I guess, to beat aging, focusing on the brain, and hopefully other people will take care of other parts of the body. But my motivation, I guess, is just life. I think it beats the alternative, right? I enjoy life. I like I want to continue. I want to have the people that are close to me continue, and have everybody else have the opportunity to do that as well.
Cool. I don't disagree.
Yeah.
Thanks.
Thank you.
A quick question, a quick answer, but a very important one.
Yeah.
And I think you answered that quite well. You said this in the book too at one point. Your closing sentence: "If we love life, we should be free to live it for as long as we can." That's a nice way to close out a book like that. So, All right, we're 20 minutes past. Just let us know how we can keep doing this for. I think our record time was over 3 hours for one particular call. So,
Yeah.
all right, Laura Minkini, welcome. Maybe a minute, about a quick background on and your question.
Hi, Rob. Nathan, thanks for having me on stage. I just came to say a hi to Jean because I really like him. So, I'm a big advocate for longevity. And by longevity, I mean how to match our health span to our lifespan. And the reason I got into longevity was because I think we need to change the current paradigm of aging. It's not quite pleasant. A lot of people are spending too much time sick, and the quality of life is really poor in the last days of the last years of our lives. I just wanted to say to Jean that one of the biggest things, I mean, in a lot of studies, but the one thing that people are always afraid of, and in terms of aging, is losing their minds, meaning their brains. So what he's doing is so important. And I just have a question in terms of the work that he's doing. John, do you think that within a 10-year span, you will have advanced a lot of the work so that we can really regenerate the brain?
Yeah, I really hope so. And Laura, thanks for your question. And I'm a fan of yours and what you're doing as well. And even though I read your tweets, even though I don't often respond. um, but yeah, I mean, this sort of gets back to the difficulty in predicting a timeline for success, even just for the brain. Um, my dad keeps asking me the same thing. When can I benefit from this? Um, and you know, I hope it's in time for him. I don't know, um, but it's not impossible. So we've set again with the proper backing, um, where we have our teams in place and we're progressing. We have an optimistic time to clinical trial for our first case study, which would be stroke, for example, or local brain damage. Could also be trauma, but stroke is just the most common form of brain damage, and so recruitment certainly wouldn't be a challenge. And so our optimistic, if we have all our operation running in parallel, all the aspects of the project in parallel being developed, we can get to clinical trials, I think, maybe in 5 years for stroke. And then, the next thing, it would take at least 5 years, I think, between that and the next application, just because the time it takes to initiate the trials and also to get at least preliminary results before moving on to another trial. But the next trial would be much closer to something akin to aging, like frontotemporal dementia, for example. So it's a little less localized, but it's still not, the whole brain, and so can be addressed with these new cortical replacements. So it's not impossible in 10 years that we could be at that point where we're tackling that, which is much closer to aging, because it's a form of dementia. And then after that, it's just, if that is shows success, then we move on to senility, which essentially is aging. But again, that's with everything operating at an accelerated pace. Um, at the speed we're going now, there wouldn't be a lot of us would not benefit from this. So that's why we need to accelerate this. But it's not impossible within 5 to 15 years to be showing benefits in humans of these types of transplants. Transplants are already approved in clinical trials for a lot of different brain disorders, degenerative brain disorders. The idea is not repair, though. They're just putting in cells that provide good factors for existing tissues. So what we're doing is a little different, a little more ambitious, but I think we can make a convincing case given that there's no alternative approaches that are working. Thanks, Laura.
Thank you. One more question really quickly. How are you finding the subjects for your trials?
Well, we haven't started the trials yet. We haven't. We're optimistically 5 years away from that. But we foresee that the size of damage due to stroke will be an important inclusion criteria, because we don't want to start with strokes that take out half your brain. We want to start with something a little smaller. The age might not matter so much. We'll probably be open to different ages, but as long as it's not like a 95-year-old where the risk of succumbing to the surgery itself would be too high. Um, so, there'll be some exclusion criteria possibly for very advanced age patients. But yeah, I don't know if that's what you were asking, but yeah.
No, I'm just curious if when doing research, it makes sense to look at people that would have the propensity to have degenerative diseases because of their family history.
Oh, I see. Yeah, yeah. So when we get to more widespread damage, application to more widespread damage, yeah, that's definitely a possibility. Yeah. It actually makes scientific sense, right? Because
I would think so.
yeah, we know what the outcome would be without treatment, and so there's a better basis for comparison, perhaps.
Thank you, Jan. It was nice meeting you.
You too.
Great, great question there. Ilya Osipov, welcome.
Hey, so I apologize if this is a stupid question. I'm just a programmer, so I'm not a PhD in biology. But your approach focuses on progressively replacing neurons in the brain, and I find it promising. But my question is, could we just add more neurons to maybe like increase intelligence? Because I know there's like a 0.4 correlation with brain volume and IQ, which is maybe like surrogate marker for intelligence. So, and also like thinking about like the difference between monkeys and humans, one variable that differs is the brain volume. So, do you think we could just add more neurons?
Yeah, even in our approach, the first step might be to add, and it's not just neurons. So we're actually adding, brain tissue, so the full complement of cell types. But yes, I think you can initially add, in part because the brain atrophies as you age. And so space is created within the skull to add a certain amount of tissue or cells without having to remove anything. So yeah, I mean, the quick answer to your question is yeah. I mean, you can certainly probably add cells alone, or better yet, nice young tissue substrate, uh, and show benefits without having to remove anything over time. But if eventually, the old surroundings, as we mentioned, in response to discussion with Colin, would have to be replaced as well, because that environment will be eventually too toxic. Thanks. Yeah.
Thanks for the question. Looks like we got another here. How are we doing for time, Jean? By the way, it looks like
I'm,
Colin left us. So
I'm good. Yeah.
okay. So for anyone that just joined in the last half an hour or so, we're having a conversation here with Jean Hébert, who is a professor and researcher at the Albert Einstein College of Medicine in New York, and wrote a book called Replacing Aging. Fascinating conversation, incredible book, very thought-provoking. Just a reminder for anyone that comes on stage that we're recording this, so if you come up, you're consenting that we can use your image and voice in the recording. So with that, Bruno dos Santos, welcome to the stage.
Hey everyone, thanks for having me. Hello, Jean. I am
Hi, Bruno.
halfway through your book. It's very interesting, and after this, I definitely want to read some more. I work as a software engineer and one of the founders of a company in Georgia called Ginfinity Precision Medicine. We work with biomarkers of aging and providing assays to consumers. And my question to you is, what is the current landscape like commercially for regenerative medicine? Are there any unmet needs that the private sector can help with pushing, replacing aging along?
Yeah, I mean, I think it's a growing landscape, especially in the longevity space. So I've been talking to a lot of people in the private sector, and although it's still underrepresented, I think the interest is growing. Um, uh, there's companies working on lab-grown kidneys, for example. So there's a tremendous need for kidney replacements. Um, and so there's definitely growing interest. A lot of it is sort of um, not yet ready for at least startups to, to or investors to be willing to fund as startups because the uncertain about the turnaround time. But there are a lot more cell-based companies now, although a lot of them, there have been some in the past too that have failed, a fair amount of them. But again, as time goes on, the technology is getting better and better, um, and more of these are popping up. So, you know, I don't know if I'm answering your question, Bruno. Maybe you can tell me if I am or not. Um,
Yeah, I definitely do agree with you that I believe there is interest in general in longevity. Um, but is there any value to be gained today in regenerative medicine? And one example I can think of, I was reading a few years ago about skin scaffolds for burn victims to try to replace the skin, and I think that's a pertinent example for this field.
yeah. I mean, yeah, I think definitely there. That's a good. Skin is a good example. I mean, it's been one of the earliest tissues to be transplanted on a regular basis in the clinic from lab-grown skin. Um, but there's still a lot of progress that needs to be made. I mean, the skin lacks the different layers. Like it just has sort of the superficial layer. It also lacks hair follicles and sweat glands. So there's progress to be made, and I think startups are a perfect place for that to happen. I'm not. I don't know. Maybe it's already happening for skin. You seem to be aware of this company. I'm not sure which one you're referring to, but I think that's perfect. So they have something that can be used um, already, and it needs improvement. But it's already something that can be used. So they're ahead of the game in that sense. Um, yeah. I'm not sure I'm answering your question, but maybe you can clarify. I mean, it may just be outside of what I know in terms of the companies that are out there working. I know there are a lot and we're interested in some of them. For example, a lot of the cells that we use in regenerative medicine come from these very immature embryonic cells. And so there's a company working on making sort of a universal donor cell that wouldn't elicit a immune response, for example. Um, and there's also different companies working with growing organs, human organs, in pigs, for example. Um, so yeah, I think we're getting closer and closer, but um, there's definitely interest. Yeah.
Thank you.
Yep, thanks.
Oh, just quickly, those two companies, I guess the second one is eGenesis, right? The George Church startup. And the first one with the universal stem cell, is that HX or is that someone else?
Yeah, I don't know if they're the only two, but yeah.
Okay, yeah, I think Revivicor is also pursuing xenotransplantation,
Yeah.
but yeah, very cool. Thank you.
Not necessarily good for the brain, though. A little more complicated.
Yeah, definitely.
I think there's another question.
Yeah, Nathan, you want to take over for a minute? Just take a quick
Yeah, totally.
take notes.
So Ilya, do you have a question?
Yes, how you doing? So I'm gonna have to apologize. I joined late, so I missed most of what you've mentioned. But I googled Gene's profile. I'm on einsteinmed.org, and I looked through it. So I think I'm a little bit more familiar now. I'm coming from a slightly different perspective as a parent of a child who has a neurodegenerative disease, and we've done MRIs, and we can see that we do lose some parts of the corpus callosum, parts of the brain over time as degeneration takes place. Now, we are working on treatments gene therapy treatments that will, in theory, stop that problem. However, we've lost number of neurons or part of the brain matter, I guess, as it took us to develop this treatment. So my question to you is, I guess the first question is, what do we know about the brain? Does the loss of the brain tissue directly equates to a lifelong problem, or can brain compensate and learn to use different areas of parts of the brain for compensation? And two is, can we apply what you're doing in children with diseases that I'm thinking of?
Yeah, I'm really glad you asked that, because I think that's a very important point that I should have emphasized, and is definitely worth emphasizing, is that the applications for this technology are not limited to just stroke or longevity or age-related, um, brain disorders. And so, yes, absolutely, this, if anything, would work even better in the younger the individual, right? Because there's already more plasticity. The environment is better for incorporating new tissue in general. So absolutely. In terms of how the brain works, I mean, this is still something that's being figured out. But we do know that that the younger you are, the more plastic you are, right? So there's examples where uh, young individuals had lost half their brain due to some developmental disorder, and that wasn't even diagnosed until they did an MRI or something to see that that half the brain is missing, because otherwise they behave fairly normally, right? So again, this is due to the ability of the brain to compensate, which is based largely on plasticity. You use what you have for what you need. Um, and again, this is something that usually younger individuals are better at because their tissue is not yet mature, and so it will adopt the functions that are needed or useful to the youngster much more easily than the adult, where I gave the example of language early on, which you may have missed, but even in adults of advanced age, language over time can move to a completely new substrate. This, of course, can happen a lot quicker the younger you are. So I think, is it a lifelong problem or is it compensation? I think depending on the age, it's not necessarily a lifelong problem or deficit, because if the younger you are, the more you've already compensated. But at any age, I think you can compensate, and plasticity can allow you to either regain function if you suddenly lost it, or to adopt function from an area of the brain that is slowly failing. But thank you for your question. Yeah, and I hope these either the genetic gene therapy or the cell replacement therapy will benefit your child before too long.
Thank you. If I could ask another question, if nobody else has a question. So uh, this is just so I understand. Right now, it's pretty straightforward to take an iPSC of a person and then create. Supposedly, you can create neurons of different kinds.
Right.
Uh, the, the, if you were to inject the neurons that are created in the lab, and I think you mentioned that there are a few companies that are already working on something similar. I guess where is the challenge? Why isn't that so simple to take neurons that you've created in the lab and literally inject them into a brain?
Yeah. So, you know, in some places it might be more simple. For example, in Parkinson's disease, in that case, there's a particular type of neuron that's lost, the dopaminergic neurons, and you know the transplants have been occurring for that type of neuron for since the 80s, and more recently from iPS-derived cells. So there's at least 3 groups that I know of that are using iPS-derived cells to generate this simple neural precursor cell type that's then transplanted, not even in the right place of the brain, but in the target area, and in the past has shown benefits, and they're working on getting that to where to be even more successful moving forward. But for the part of the brain, the neocortex that we use for higher cognitive function, it's what I'm using now to try to formulate sentences that are comprehensible, and what you're using now to try to make sense of what I'm saying. Um, so it's a more conscious part of the brain. Uh, that's a little more complicated. There's a balance of cell types that are needed. There's excitatory cells, there's inhibitory cells, and there's a lot of modulatory input that comes from other parts of the brain that needs to be reestablished. So it's basically just that the problem is a little more complex than putting in one particular cell type. Um, and unfortunately, most of those doing research put in essentially, just the main like excitatory neuron cell type, and that couldn't possibly function normally. So I think more groups need to start thinking about combining the different types of neurons that are required for normal tissue, uh, um, normal function, as well as the topographical organization of those cells, also equally important. And those that just hasn't been developed yet technically. Um, but that's what we're working on, and I wish 100 other labs were working on it too, so that we can get there faster.
Awesome.
Thanks.
If I could ask one, I don't know. I mean, I'll shut up, but I do have one more question. Let me know if I just ask too many questions.
I don't think it's an issue. I have so many questions to ask myself. Other people do, but really, the question is, John, how much time do we have? Thinking maybe we can go till 6 on the recorded part of this, and if you still have time, if anyone still has time, we can move to a new room that's more freestyle and will not be recorded. Does that
Yeah,
sound?
I can go to 6, but I have to stop at 6:15. So yeah.
Okay, let's do that then. So yeah, go ahead, Ilya.
So last question. Again, this is a fascinating topic for me. As I learn more, and I've come across a bunch of researchers, and I'm sure you know of the research where you, I guess, play with specific genes and that control the neuron growth factor, and depending on what you do with these genes, you turn them on, you turn them off. You could, in theory, let your this is an assumption, I guess, let your body create more neurons. I'm wondering if what you think of that, if it's indeed how it works, or maybe I just misinterpreted it.
No, that's also a very good question, because I think a lot of people who aren't necessarily experts in the brain assume that, and they've heard that, oh, we have stem cells even in the adult brain, that we could control these stem cells to regenerate, um, um, you know, dysfunctional or lost brain tissue. Uh, but the first thing that's important to keep in mind is these stem cells are very restricted in their location. There's a very small part of the brain uh, that is thought to house these stem cells. And they certainly do in most mammals. In humans, it's still a little controversial, I guess. I think the preponderance of the evidence is those stem cells do exist even in the adult human brain. But it's in a very restricted location. And these cells are when they give rise to new neurons, uh, they don't migrate very far at all. It's just, microns away from the place they were born. And they give rise to, again, a very specific neuron type. So, in a brain as big as the human brain, to figure out how to coax these stem cells to move to new parts of the brain and become different types of neurons is really beyond what we can do. I think the approach of just putting the correct neural precursor cell in that part of the brain is a much more feasible approach than trying to reprogram cells that are there that might have the capacity to do that. So, you're right. We use genetics to study brain development and to study what controls the behavior of these stem cells, even in the adult brain. Um, and that was very interesting, and we learned a lot. But in terms of repair, we've put all those projects aside uh, because I don't think they're going to be directly useful, or certainly not as feasible as just growing our own cells in culture that are exactly the right type already that we want and putting them where they need to be in the brain. Um, both approaches would involve some level of surgery because these genetic manipulations, you have to put something in, like viral delivery or something. So I don't know that one is even, less or more or less invasive than the other. Uh, certainly the tissue replacements that we're talking about for rejuvenation are more invasive. But in terms of just delivering cells for therapeutic purposes, I think it's much easier just to do it with a cell-based approach as opposed to a genetic-based approach.
Thank
I
you.
hope that answers your question.
I have one more question, if that's okay.
It's okay with me.
Yeah, go ahead.
Okay, cool. So obviously you just highlighted, I think with respect to Ilya's first question, that there's a lot of devil in the details in the sense that we can't just insert the neuron, like just inject the neurons and expect everything to work. There's a bunch of various ratios and interactions between the supporting inhibitory neurons and like various types of neurons in different
Right,
places in the brain.
right.
So my question is, do you think that there will be varying degrees of different types of protocols or procedures between different people? Because each individual person will probably have like differing ratios with respect to how the neurons are organized, how quickly the neuroplasticity happens, and all that. Like, do you think there will be a lot of difference between individuals, and that might potentially be like a big stumbling block? Or do you think like when you develop the protocol, do you think it'll just be quickly generalizable to most people?
I think more the latter. If you look at the brains, even across species, the composition of the cells, their arrangement, their location, their ratios. The ratios change a little more between species, but within species, they're relatively tight. So that's not so much an issue. I think where the protocols will vary a lot more is for their application. So if we're just talking about brain repair, the protocols might be very different for different types of damage and, of course, different parts of the brain. In terms of whole brain rejuvenation, I think those can be a little more standardized because this is not, we're not responding to acute damage. This is, we can establish protocols that we can follow over time repeatedly for different individuals. So yeah, I think the variability will come from the application more so than inter-individual variability.
Thanks. That's heartening to hear.
Actually,
Sure.
if I could follow that up with a bit of a hardball here. Ilya, number one here, Ilya Osipov, just asked, about these technical details that are nitty-gritties that can maybe complicate matters. In your book, you mentioned at one point that the actual, like mechanistic basis of memory in the brain is still being figured out.
Right.
But there's evidence that memories are contained in or essentially encoded in like networks of thousands of neurons, et cetera. And if they're placed in a gradual way, they can be preserved. Especially if the person is being, and you said this in the book, if the person is being reminded of the memory as this is happening or around the time after it's happening. So is there a risk that maybe that's not going to work as effectively, and it will lead to physical longevity, but the person will lose their memories or become a different person as the replacement occurs?
Yeah, I mean, that's definitely a consideration, right? But that's true regardless. If we don't touch the brain, you're going to forget things already, right? If you don't use them. And so, in that sense, it's not that different, whether we're doing a manipulation or not. And eventually, you're going to lose more and more of your memories with age, right? So, but it is an important point because the pace at which I mentioned earlier, that you start to do silencing of a brain area at the same time as you're providing a new substrate to allow plasticity to do its thing and the function to seamlessly move from the old part of the brain or tissue to the newer tissue. The rate at which you do that will be very important, right? You can't go too fast because we know it takes, at least 3 years for a function to move. But these details will need to be worked out for how fast you want to do that. Um, too fast, and I think I might have mentioned this in the book as well. You become more like an infant, where you can learn things really easily, but you may forget some of the things that are important to you. And too slow, then you're not going to outpace aging itself. So that rate needs to be determined, and there might be differences in the rate of replacement that individuals are comfortable with. And there's also, different parts of, even again, if we're just focusing on the neocortex, there's primary neocortices that are very specialized in their function, and then there are associative parts of the neocortex which might be a little more important to us, that we might want to replace more slowly and very carefully. These are the parts that we use, like the frontal cortex, the frontal lobe, temporal lobe, right? These are where we have our deeper, more complicated thoughts, where we integrate all our input, our perspective. We plan things. So the rate might vary for the different parts of the neocortex as well. But as we know from examples in humans of advanced age, you can lose a substrate underlying language without losing language, right? Because if you do it slow enough, it'll move to a different substrate. So again, language is a big one. And if, so I think it's something we can do.
And about memories, I mean, it's one, suppose you can do this and just general memories. I mean, if you do this kind of replacement, but then you lose all the memory of the first, let's say you do this when you're 60, right? And then the replacement occurs and you just can't remember anything from the first 50 years of your life. So that leads to a strange kind of,
Well,
life extension where
that would mean that the protocol was not working, right? Because that's not how it should work. Um, you know, the memories are not lost if they're moved to a new substrate, a new circuit, right? Just like the example with language, you never forgot how to speak, right? You never forgot the meaning of the words, right?
this is true, but language and general memory, are they really equivalent?
No, not necessarily. There are non-language based memories, but the principle of plasticity is the same. If it can work for language, it can work for other functions as well, other memories.
Because language is something that you use constantly, whereas memories in general are something that you don't quite as constantly exercise, right?
Right. So, no, I mean, it's true that if you don't use it, you will lose it one way or another. Whether you do these manipulations or not, you will forget things that you're not reminded of. You're forgetting things now. I'm forgetting things now because we're not using them, right? So that there's a risk of accelerating that, um, but again, that can be mitigated by you know uh, focusing on the things that matter to you and remembering them and using them.
So write things down and take pictures and remind yourself of everything that you want to remember. Okay, so we're almost at the 2 hour mark.
Just living life and doing the things that you like to do normally will allow that to happen more naturally. Yeah.
Absolutely. So you said you can go for another 15 minutes almost. So instead of moving to another room, maybe we'll just keep it running till 6:15 Eastern here. But we can just open it up to some more open-ended questions as we would do in the open-end chat. So maybe I'll start here. Jean, your book, like I said, is the best thing I've read on this kind of general theme since Eric Drexler's Engines of Creation, which is almost like science fiction. So my question to you would be: what books, films, or the like had the most influence on you, or you find most interesting and thought provoking in that way in the science fiction genre?
Yeah. It's been a long time since I've read a book for fun. A long time. Um, yeah, I don't think they were relevant to my, what I work on. I just think
Do you watch? Are you a fan of Futurama?
no, I don't know what that is. Sorry.
Okay, well, maybe I'll send you a few clips from Futurama where they have effectively a brain in a jar. Uh,
Okay. Yeah, yeah.
so it's comedy, but they foresee a future in a comedic sense where these things might be possible.
Sure.
But I was asking more, conventional type of books. But if fair answer, if you don't have anything particular in mind, but
Yeah.
um,
Right. I mean, in terms of what helped shape a bit my trajectory in terms of science was another science book written by Caleb Finch. It's called, I think it's Longevity, Senescence, and the Genome, or something like that.
yeah,
Uh,
you reference it a few times in your book.
yeah. I mean that's also a good reference in terms of getting a really broad perspective on the question of aging. I mean, by senescence, you know, the term in that case is used to mean aging and not what it's now come to mean is the non-proliferative state of cells. So yeah, it's a very broad perspective. I remember reading that book, and you know, it was at the time was super thought provoking for me, and helped guide my thinking ultimately in terms of where I am now. That replacements are the way to go.
So longevity, senescence, and the genome for anyone listening, and
Yeah,
I'll include myself in that amongst the listeners here. Actually, that brings me to
yeah.
another
But
thing.
since we have new listeners, I'll reiterate that paper from about a year ago from Alexander Fedintsev and Alexei Moskalev. I think that's a must read as well. It's much shorter as well than a book, but yeah. And that's in Aging Research Reviews. That's the name of the journal.
So we're 2 hours in here. We've got 15 more minutes with Jean Hébert, professor and researcher at the um, Albert Einstein College of Medicine in New York, working on replacement-based therapies for largely for the brain, but talks about the whole body as well. Fascinating conversation we've had here. This is being recorded, so we're going to put that up on YouTube shortly. Nathan, I guess maybe you'll close out in a few minutes, but Jean, before, and again, if anyone else has questions, just speak up or raise your hand. Jean, who would you like to see? Who would you hope that we can potentially get as a guest on a future episode? Since you brought up Caleb Finch and how influential he was, do you think he might be an interesting person to ask about these things, or who would you have in mind?
Yeah, I mean, I think he might be an interesting person. Let's see. Uh, who else? You've already had Aubrey. Um, I mean, I have to get back on you on that.
Okay, no pressure. Just
Yeah,
just putting it out
yeah,
there because we like to ask the guests as well, because
yeah,
the point of these conversations is to try to you know uh, exchange ideas, and I mean one of the points is to try to exchange ideas amongst people in the scene, and it's interesting to hear who the who you would want to. We've gotten some interesting responses too. Like Aubrey's immediate answer was. President Biden, and we had a guest previously that mentioned Bill Gates also. So
yeah.
uh,
Excellent. Yeah.
yeah,
So
take your time and get back to us when you can.
any potential large philanthropist would be good to get their perspective. And to that, in terms of a scientist, I might add somebody who's actively working in an area of regenerative medicine. The biggest name might be Anthony Atala at Wake Forest. Um, I think he was the keynote speaker at one of Aubrey's conferences, and it'd be great to hear where things are at in terms of his progress, because they're working on many different lab-grown organs. But that might be a little self-serving given my interest. But again, I think that should be of general interest to your audience as well.
Yeah, absolutely. We'll see what we can do there. So, folks on the panel here, if you have anything else you'd like to ask for the next couple minutes, now's a good chance, and then I think we'll try to close this out.
Yeah,
John
so
looks
I
like you have a
actually,
question.
yeah, I have two more questions, but one should be quite quick. So most recently, my background is in artificial intelligence, and I was just wondering if you had any plans to use machine learning, deep learning, or AI in any of this brain replacement research.
Yeah, I mean, I'm really excited about incorporating AI in certain steps, right? I mean, it's being used more and more just in terms of assessing large data pools, which we will also be using in measuring how well our graphs are working. But beyond that, even in we're going to need pretty smart machines, I think, in terms of executing some of these graphs in the human brain, which is a little um, more topographically challenging than the very flat rodent brain, for example. Um, so, we'll need machines that can recognize layers within tissues and then, deposit cell layers that match surrounding layers, for example. I mean, this can be done by hand, but, I'm hoping that advances in AI will facilitate that. And then, as we've touched upon, there's the brain-machine interfaces, which are getting better and better. Um, those are very active fields, and so I expect those will be very good, and will also provide potentially useful applications to the in terms of complementing the grafting approaches. Yeah, you had
Yeah,
another.
great. Um, yeah, yeah. Yeah, I mean, I think there is a lot of potential and opportunity, and it's good to hear that.
Yeah.
I guess at least people in the domain are thinking in that direction, how to incorporate AI into their workflow somehow. But
Well,
my second question,
that'll be your job, because I wouldn't know the first thing about how to develop these, but I have some ideas of how they can be used.
yeah,
So,
that's
so,
definitely,
so I hope people like you develop them with us in mind.
yeah, definitely something I'm thinking about, and I think hopefully other people are too. But my second question goes back to something that you and Robert were talking about, going back to this idea of gradually silencing parts of the brain. I think that's very interesting in trying to recreate what happens with a glioma. In some ways, a very important aspect of this brain replacement therapy. And I don't know if you have specific strategies for doing this, or ideas in this regard of how you would actually go about doing the silencing, because it seems like quite a difficult task in my mind.
Yeah, no, that's a very good question. There are different ways to silence, you know, CNS tissue or brain tissue. There are physical ways, just with temperature, cold temperature. There are pharmacological ways where you can silence neurons, but those are hard to control, in terms of space. I think a much better approach is to use techniques that are somewhat more recently developed, um, where if you deliver uh, channels to neurons in a particular area, that then using penetrating red light, you can silence the neurons that now express this channel. And you can do that part completely, much less invasively. So, you still have to remove maybe a bit of the overlaying tissue, but without touching the brain itself. And that light, you can adjust that light in terms of the area over time at any rate you want, right? And it's sort of a laser light, so you can very, uh, you know, um, precisely engineer the silencing of a brain tissue that way. Um, and I foresee something like that being the best approach. The technology, by the time we get to that stage where we're applying this to reverse brain aging, technology may be way better. There may be better options, but right now, that looks like the best technological approach. But it certainly, again, already seems like that should work, right? So, yeah, thanks. That's a very good question.
All right, I guess we have time maybe for one more question. Ilya, go ahead.
Okay. So, Jean, supposing we like use your strategy to replace, let's say, 20% of the of old neurons in a brain. So we have 20% of the neurons are young, and then the rest of the 80% are like still old. So my question is, would the molecular damage that is in the 80% old brain, would that damage dilute such that eventually the damage is uniformly distributed among the brain? Or would that damage just kind of remain in the old part of the brain and not really move to the new part? Because if it's true that the damage would dilute, then that would suggest that you don't actually have to replace the entirety of the brain. You would just have to replace enough of the brain such that the damage would dilute to a point where the overall amount of damage is below the pathogenic threshold. So, in essence, the question is: do we have to replace the entire brain, or do we just have to replace enough of it so that it dilutes?
Yeah, no, the damage wouldn't dilute. A lot
Oh,
of this damage is in extracellular matrix, and it's the outcome is a stiffening of all the tissues in your body, but including the brain. So the brain essentially, you even get calcification in the old brain to some extent. So it just, it's less fluid. There's much less opportunity for dilution. Things are sort of becoming frozen in place with these cross links and damage over time. So the damage does not move, and that's for extracellular. Intracellular as well typically does not move. There are exceptions to that, like prion disease, where damage can move from one part of the brain to another, but in that case it doesn't dilute; it just spreads, which is not good. But in general, no, you wouldn't get dilution. Yeah.
okay, thanks.
Thanks for that. Great question. Nathan, I guess we can close it out here. Nathan, you still there?
Yeah, of course. Okay. So thanks again, John. This was a fascinating conversation, and obviously a great honor to have you here. And so maybe just a few closing remarks. So number one, next week, our guest will be Kristen Fortney. She's the CEO and co-founder of BioAge Labs. So that's a machine learning and AI drug discovery company that's targeting aging. And then number two, so just to reiterate, Jean, his project, he mentioned that he might be hiring some people to for this brain aging reversal project. But also more importantly, if anybody in the audience is interested in helping in terms of funding, if you know someone who is maybe a philanthropist or you know some sort of source of funding, that would be great. You can reach out to him. Just find his email at the Albert Einstein College of Medicine website. And I guess the third thing, go buy his book. It's an incredible read, Replacing Aging. It's not too long either, and it's just got some of the most interesting ideas and just like the most, I would say, the most fundamentally sound approach to completely addressing aging versus maybe some of the other things that we're doing in the longevity industry, which I think are still very cool in terms of slowing aging. But yeah, definitely one of the most interesting ideas out there. So with that,
Great.
Robert, is there anything else you want to add, Robert?
No, we didn't get to do the usual lightning round questions. So we still got like 2 minutes. If you want to pop them off, then we can close the recording.
Okay, sure. So just last sort of quick questions, lightning round. So John, number one, how long do you want to live?
Yeah, I remember that question from your previous that you posed to your previous guest as well. I don't know. I don't foresee life becoming boring. I think there's always ways that it becomes interesting. Especially, we can't imagine what the future has in store. So, I would love to see as much of it as possible.
Awesome. And number two, cryonics. What do you think? Yay or nay?
It's I guess it's better than decomposition, for sure. But I just wish that they were doing faster development in terms of not so much the vitrification, but the reanimation part, right? I know groups are working on this for organ transplantation to preserve organs for longer so they can get to the patients that need it. Um, so they're working on this, but there again, not enough investment in that area as well. Um, so I think right now I don't know if it works or not. Well, clearly there is no way to reanimate tissue with the current protocols of vitrification. I guess people are hoping that we'll figure that out later. Um, but yeah, anyway, those are my thoughts on cryonics.
Okay, great. And then last question, this is kind of an out there question, but the Fermi paradox. Why do you think we haven't encountered alien species?
I think it's mainly technological. We don't have the tools to see enough. I mean, and the limitations of space, basically. It inhibits us from seeing anything but distant past. This, the light that hits us now from places far, far away are from their distant past. That doesn't help, and our inability to travel uh, very far beyond planet Earth is also a tremendous limitation. Um, but yeah, I mean, like Carl Sagan says, billions and billions of stars. It's hard to imagine that we're the only life form out there. So I think at some point, that would be a great reason to continue living as well, is to start encountering life forms on other planets.
Maybe the life forms figured out how to live forever and said, we've had enough. But that's something that, kind of Douglas Adams-esque. And on that note, I have to read that book. But thank you so much, Jean, for your time.
Yeah,
Amazing book, amazing conversation. I think on my part, this is one of the most interesting ones that we've had so far. Really hope we can have you back, perhaps with some other experts to continue the conversation, maybe debate some of the ideas, the directions that the field is taking. Nathan,
that'd
thanks
be great.
for inviting Jean over here. And thanks everybody for being patient with us and sticking around. We're going to have the recording up on YouTube. Be sure to follow us on the app, Twitter. Follow Nathan's newsletter for future updates and information on the scene.
Yeah.
And with that, Jean, I guess we'll close out the room. Thanks
Yeah,
so much for your time.
thank you very much, Nathan and Rob, and all the participants. I appreciate the opportunity to share some of our ideas, and yeah, I look forward to a debate would be great with other perspectives on longevity and how to achieve it. That'd be great.
All right, we'll try to arrange it.
Yeah, thanks a lot.
Okay, thank
Okay,
you.
thanks. Gonna close out. I think as soon as John leaves, the room might close. So because he opened it.
Okay.
Looks like it's still around. All right, let's close it out.